Hello and welcome to the webinar today entitled AAV Symposium, Characterization and Biodistribution in Gene Therapy. My name is Miguel Medina. I am an account executive with Advanced Cell Diagnostic, a Bio-Techne brand, and I will be your moderator for today's event. I'd like to thank everyone for attending, and I'm happy to welcome our featured speakers for today, Dr. Chris Bunker and Dr. Chris Hager. So we'll go over the agenda. Following the introduction, we will have a 60-minute presentation divided into two segments. The first segment will feature Advanced Cell Diagnostics RNA in situ hybridization technologies, RNAscope and BaseScope, which can be used to visualize, detect, quantify AAV and lentivirus vector biodistribution and transgene expression in preclinical animal models. In the second segment, we'll explore the various capillary electrophoresis techniques offered by ProteinSimple that can be easily implemented along the development path to support gene therapy. Following the presentation, we will have a 10-minute Q&A session with the speakers and Bio-Techne representatives. This webinar is supported by Bio-Techne. Bio-Techne brings together the prestigious life science research brands of R&D Systems, Novus Biologicals, Tokris Bioscience, ProteinSimple, Advanced Cell Diagnostics, and Exosome Diagnostics to provide the scientific research community with a comprehensive and world-class product portfolio of reagents, media, extracellular matrices, small molecules, assays, and instruments. Before moving into the talks by our featured speakers, I have just a few housekeeping items to address. Throughout the webinar, we invite you to submit any questions you have for our presenters using the Ask a Question box in the GoToWebinar control panel. These questions will be asked to the presenters directly following the presentation. In addition, right above the Ask a Question box, you will see a tab called Handouts. In the tab, you'll find extra content that relates to today's webinar. And now I'd like to introduce our first speaker, Dr. Chris Bunker. Chris Bunker is the Senior Director of Business Development at Advanced Cell Diagnostics. Dr. Bunker did his doctoral research in biological chemistries at Harvard University and the Leukemia Society of America postdoctoral fellowship in molecular genetics at the University of California, San Francisco. He was a senior scientist at Genome Therapeutics before joining Cell Signaling Technologies at its inception in 2000. Dr. Bunker received an MBA from Boston University and then joined Advanced Cell Diagnostics, where he has led its assay services business for the past 9 years. Chris, the floor is yours.
Hello, my name is Chris Bunker. I'm the Senior Director of Business Development at Advanced Cell Diagnostics, which is part of Bio-Techne. My focus at ACD is on our professional assay services business, and today I will speak about our work in support of AAV-based gene therapy development with assays that enable morphology-based gene therapy, biodistribution, and expression analysis. Our services utilize ACD's RNAscope, BaseScope, and microRNAscope in situ hybridization technologies, which provide a means of visual and quantitative analysis of essentially any endogenous or transgene mRNA or microRNA, as well as providing a means of detecting AAV vector DNA in routine FFP tissues. Our BaseScope assay in particular has been important in that it provides the only tissue-based analysis method to differentiate human transgenes from endogenous animal orthologs. The phrase morphology-based biodistribution was coined by Asaf and Whiteley in their review article on considerations for preclinical safety of AAV gene therapy products. In their words, the addition of morphology-based methods such as in situ hybridization for assessment of AAV nucleic acids may be critical to understand the cellular distribution within a tissue and provide unique insight that can influence safety and efficacy considerations. For our part, Advanced Cell Diagnostics has tools that are uniquely able to provide this type of morphology-based analysis of gene therapy nucleic acids. First, by way of introduction to Advanced Cell Diagnostics, we are again a part of Bio-Techne. We have about 200 employees in the San Francisco Bay Area. Our products and services are provided throughout US, Europe, and Asia. And our products are primarily probes. So we have nearly 25,000 probes in our catalog for the detection of essentially any RNA in fixed tissue, whether it's mRNA, long non-coding RNA, splice variants, even point mutations or microRNAs. We have probes and technologies for detecting any one of these. Our probes are provided either as products, kit-based products, or through use in our assay services. I think it is fair to say that our Pharma Assay Services group has greater expertise in RNA in situ hybridization assays than any other CRO. And in 2020, our Pharma Assay Services have become increasingly important to our customers as many of their labs have been reduced in their capacity. Our capacity to run RNA-scope assays has not been impacted, in part because we have nearly 20 Ventana autostainers and Leica Biosystems autostainers running assays nearly 24/7 with minimal person time involved. In addition to these assay services, we also provide tissue sourcing and basic histology services, digital slide scanning, and data analysis using Halo image analysis software from Indica Labs, and scoring by board-certified pathologists. So our technology is called RNA-scope. However, it is able to detect both AAV DNA, genomic DNA, as well as transgene mRNA. In the context of the types of tissues that gene therapies are being applied, retinal tissues, liver, brain, in all cases, the viral nucleic acid that's being delivered and the transgenes that is being expressed from that vector can be detected with probes, either to the transgene sequence or its corresponding RNA, or probes to non-coding regions, promoter regions, or the antisense strand, which is also not transcribed. These assays provide us with a means of visual assessment on a single cell level in intact tissue of both vector copy DNA as well as the transgene expression from those vectors. The RNA-scope method has three key features. The first is that it's intended for and enables detection of RNA nucleic acids in formalin-fixed parapermitted tissues. It has the ability to detect and then quantify single molecules of RNA in fixed tissues, and it has very high level of specificity, much like any extraction-based nucleic acid assay. It's slide-based like IHC or immunofluorescence, but in this case, we're hybridizing with oligonucleotide probes, which then, through a series of amplification steps, will localize signal to the point where a visible dot will appear for each individual molecule being detected. A chromogenic dot in the case of HRP or alkaline phosphatase assays, or fluorescent dots in the case of multiplex fluorescent assays. The RNA-scope probe design is such that signal is dependent on paired oligonucleotides hybridizing to the target sequence. A typical RNA-scope probe will consist of a large pool, about 20 of these pairs, tiled along a region of about 1 kb of sequence. Each one of these pairs is able to generate signal. So by incorporating a large number of these oligo pairs into a given probe, we build in a high level of redundancy to ensure high efficiency of molecular detection. Every single one of these oligos is tested in silico to ensure that it will hybridize only to the intended target sequence and not cross-hybridize with any other sequence in the sample. RNA-scope and base-scope in situ hybridization technologies are the primary assays that we use in our assay services. RNA-scope is the assay on which ACD was founded 10 years ago, and it meets the needs in most cases, particularly for looking at and detecting endogenous transcripts. In the context of gene therapy, oftentimes we'll perform duplex or multiplex RNA-scope assays to simultaneously detect AAV transgene mRNA, AAV vector DNA, and then with fluorescence and higher plex, we can incorporate cell marker probes to identify cell type. We can also incorporate either immunofluorescence or IHC as another means of identifying cell type in combination with RNA-scope probes. The base-scope assay has been particularly useful and valuable to many of our gene therapy studies, particularly in cases where the transgene that's being detected is of human origin, and the intent and need is to detect that without cross-hybridization to endogenous mouse or non-human primate orthologs. The probe chemistry of BaseScope is different from that of RNAscope, and it allows for a differentiation of very highly homologous sequences based on using chemistry that's of higher affinity. It also uses a larger amplification module, which allows us to target short sequences, and this has been useful in many of our studies in cases where we're, say, looking at exon-exon junctions or exon skipping events mediated by exogenously delivered nucleic acids, or even point mutations that result from base editing as another therapeutic approach. The third RNA in situ hybridization assay that we have to offer is for detection of microRNAs, siRNAs, and ASOs. This was developed just this past year in 2020, and as the name implies, it is intended for detection of very short nucleic acids, originally intended for detection of microRNAs, but has been a very useful tool for detection of exogenously delivered siRNAs and ASOs. More recently, we've been doing studies to detect these short nucleic acids expressed off of AAV vectors. And there again, as with RNAscope and BaseScope, we can use assays to detect both the vector expressing these RNAs as well as the RNAs themselves. Like the RNAscope and BaseScope assay, it uses this similar amplification methodology, which enables a very highly sensitive and specific method of detecting these short nucleic acids. Here I'll show schematically how signal is amplified with this paired oligonucleotide approach. With oligos hybridizing to a target transcript, they will then be able to stably hybridize to this preamplification oligonucleotide, which then hybridizes to amplifier oligonucleotides, which then hybridize to either chromogenically labeled or fluorescently labeled oligonucleotides to generate signal. And this assay, again, allows for localized amplification that is on the order of 1,000-fold per pair of oligos, or over 5,000-fold for paired BaseScope oligonucleotides. And this happens in a way which is, again, dependent on multiple oligonucleotides hybridizing simultaneously to a target sequence, which is how we're able to generate such high levels of signal without any background signal coming from non-specific binding. Results in sufficient signal to see each molecule as an individual dot. That's illustrated in this data here from the first publication with the RNAscope method. In this case, we're showing an example of two experiments, one with HeLa cells and an RNAscope assay, in this case to detect the HER2 transcript, and in the other, a plate of HeLa cells where RNA was extracted and solution-based measurement of HER2 transcript was performed in the context of in vitro transcribe methods that could be used as a standard curve. In both cases, the number of copies per cell for HER2 is approximately the same, 14 dots per cell in the case of the RNAscope assay, and about 17 transcripts per cell from extracted RNA. And this is simply to make the point that an individual dot corresponds to an individual molecule being detected. Now, if this assay is run under slightly different conditions, which enable permeabilization of the sample and exposure of DNA, this same probe, which is detecting HER2 RNA here, reveals two dots in those same HeLa cells when we're now detecting DNA. So this is the same probe being used under conditions which expose genomic chromatin, and this assay is now showing two dots, which again emphasize the fact that a dot corresponds to a single molecule, whether it's an RNA molecule under an assay condition that's intended to detect RNA, or as shown here, the same probe detecting two genomic loci in HeLa cells, which are wild type for HER2, and multiple loci in the case of SKBR3, which is amplified for the HER2 locus. The next few slides will illustrate examples of the sensitivity achieved with RNAscope and how that compares to other methods like IHC and qPCR. We do like to emphasize the fact that a few dots matter most often, and that that's what we can detect with the RNAscope assay. In this example, this is an image of an RNAscope assay performed on a mouse lung, where the probe is detecting the RO1 expression in pulmonary vein endothelial cells. This was in relation to a CAR T-cell program, and it was this level of expression which, when these mice were treated with anti-RO1 CAR T-cells, resulted in pulmonary toxicity. and that correlated with infiltration of CAR T cells as detected by RNAscope in the lower right, as well as corresponding activation of T cells based on granzyme B expression. This was a safety assessment study which proved or demonstrated that a low-level expression of RO1 detectable by RNAscope and undetectable by IHC led to a phenotype that related to the activity of CAR T cells targeting RO1 expression. Another illustration of RNAscope sensitivity is shown here in relation to qPCR. This was for a clinical biomarker assay where NRG1 or heregulin had been identified as a marker that would predict patient response to an antibody therapeutic. Now, there were two factors that contributed to RNAscope being selected over qPCR. On the one hand, it was more sensitive when the sample was a very small needle biopsy. There was not sufficient material to get a reliable qPCR assay from that sample. However, because the RNAscope assay is measuring expression in individual cells, even from a small biopsy, there was sufficient expression information to be able to predict patient response. Moreover, it was the expression in tumor tissue which was most predictive of patient response. And that information could not be gathered from a qPCR assay, which would be a combination of expression that might be coming from tumor cells or stromal cells. And finally, in relation to nanostring here, we use the data shown here to just illustrate the additional information that comes from a visual analysis of nucleic acids. Nanostring is an extremely sensitive assay suitable for RNA analysis in fixed tissue. However, it still provides an average value of expression across an entire tissue. And as illustrated here with these three examples, TNF-alpha, IL-1 beta, and CTLA-4, what we can see if we look visually is that there are very few cells that express these markers at fairly high levels, whereas the numbers shown at the top indicate that overall the expression throughout the entire tissue is extremely low. So the benefit of having a visual assay for these markers, or for instance for AAV vectors or AAV transgenes, being able to identify the number of cells that are expressing a marker and then measuring expression within those cells is the information that comes from in situ analysis as compared to extraction-based methodology. So now back to the primary focus here of methods for looking at AAV DNA and transgene expression. Now our assay is called RNAscope because it was originally developed prior to the development of gene therapy methods, and the intention was to detect low copy endogenous RNAs. What we've observed though is that probes to transgene sequences will of course detect the transgene mRNA. In addition, if we design a probe to a non-transcribed region, either a promoter region, or if we target the negative or antisense strand, that probe will also detect the AAV vector DNA. So we have assays where we can incorporate probes for both transgene expression mRNA as well as vector DNA in the same assay. And these assays can be performed in any tissue to provide biodistribution analysis of vector genome as well as expression. And I'll use the next slide just to illustrate a few examples of that. So here's an example of an RNAscope duplex assay where we did combine probes for vector, in this case the CBA promoter probe, chicken beta actin promoter probe, is detecting DNA signal only, and the GFP probe is detecting transcript. This is in non-human primate eye, retinal tissue, and you can see obviously very high level expression. This was local administration of the virus, so there's very high copy of both virus and transcript. A little bit higher magnification here, you can clearly see some nuclei which have very strong staining with the CBA promoter probe. That's of course again genomic DNA. And then other cells where you see both GFP signal for the transcript as well as the nuclear CBA signal. So the ability to identify which cells have vector and which cells have both vector and transgene is the kind of information which comes from a duplex assay like this. Here's another example where we're looking again at non-human primate eye, retinal tissue, with a probe to an AAV transgene for Cas9. The signal is only observed in the photoreceptor layer. Presumably that relates to tropism of the virus, but here again, the ability to see that expression in a specific layer is what we can provide with an in situ assay as compared to, say, extracting RNA and measuring total amount of Cas9 transcript or vector sequence from that sample. Here is another further example of non-human primate retina, where I presume the administration of the virus was into the intravitreal layer, and as a result, there's a large amount of vector signal outside of the cells themselves. In addition, there is, of course, also vector signal in red inside the cells, and a subset of those cells also express the transgene. In this case, we're using a probe to a codon-optimized human sequence as the transgene being detected. Shown here is the other end of the signal spectrum with an in-situ hybridization assay. What's shown here is detection of an AAV vector with the green signal in a non-human primate brain. Now, in contrast to the signal we saw in the previous examples where there was local administration of the virus and extremely high signal for vector and transgene, here, presumably the virus was administered at a site quite distal from what's being assayed here. And as a result, what we see, and this is what we see oftentimes, is just a one or a few dots corresponding to one or a few copies of vector genome in just a few cells. In this case, the assay was, again, a duplex assay, but combining the AAV vector probe with one for RBFOX3, which corresponds to the new N transcript. And what was observed in this region of the brain is the fact that although there are some cells that are positive for new N in the red signal, those cells are generally negative for the vector. The vector is observed in other cell types. Here I'm showing RNAscope duplex data combining probes for an AAV transgene and, again, RBFOX3 to identify neurons in mouse brain. This is actually customer data, product customer data, showing detection with our fluorescent assay versus our chromogenic assay. Transgenes are often very highly expressed, and as you can see, the signal can sometimes be too strong and will mask the other gene being detected, in this case RBFOX3. That's where fluorescent assays can be more advantageous to capture those two separate signals in separate channels as compared to with a chromogenic assay where in some cases a strong transgene signal may mask endogenous cell marker signals. Now this data will also let me make a point about the predictable specificity of our probes. Our customer in this case ordered an RNAscope probe that had been designed against a human mRNA, which is their transgene. And what you can see in the PBS-treated sample in the chromogenic stain are a few faint pinkish or red dots, which correspond to the human probe detecting the mouse ortholog. And that is predictable across hybridization. We would have known that had we mentioned this to the customer. And that's an issue which we never face in our assay services. We would know to design a probe which would be specific for a human sequence in a mouse background. In some cases, we can do that with RNAscope. In many cases, like this one, we cannot design a probe which will not cross-hybridize with a mouse ortholog using the RNAscope assay. And that's a case where instead we would use the BaseScope assay. As I described in an earlier slide in the presentation, the BaseScope assay differs from RNAscope in using higher affinity probe chemistry, more like locked nucleic acids. It also uses the much larger amplification module, which allows us to target shorter sequences. But in particular, in gene therapy studies, oftentimes we are being asked to assay a human transgene in either a rodent or non-human primate background where the ortholog mRNAs that are endogenous to the tissue are too highly homologous for RNAscope to differentiate. And that's where we apply the BaseScope assay. As I mentioned previously, it's also applied in studies of ASO or other oligonucleotide-based exon skipping events and to look at specific splice variants or base editing or point mutation detection in intact tissues. This is an example of the BaseScope duplex assay applied for assaying AAV vector and transgene expression in mouse liver tissues. The bluish green signal appears in the nuclei. This corresponds to the AAV vector, and you see only one or a few dots in the nuclei. The red signal corresponds to the transgene, and this signal, as we oftentimes see, is distributed between the nucleus and the cytoplasm. The point of performing these assays in combination is to provide a basis for assessing how much of the transgene mRNA signal is actually based on the probes hybridizing to the vector genome. So we can see that only a few dots are apparent with the vector genome probe. Therefore, it's most likely that a few of the red dots are also a vector genome signal, and the remainder of that signal corresponds to RNA. Even though it's nuclear, again, this is something we see quite often, there will be a nuclear mRNA present that's being detected with the assay, presumably prior to its export to the cytoplasm. Another method that we oftentimes use in our pharma assay services projects in looking at gene therapy is an RNA removal protocol, which is another way in which to determine how much of a transgene probe signal is based on RNA detection versus DNA. Again, we know that the probe to a transgene will in fact detect the encoding DNA as well as the mRNA, and by applying a method as shown here, we can remove essentially all of the RNA from the tissue, and therefore any remaining signal will correspond to the vector genome. So on the one hand, we can use duplex assays of vector and transgene probes. In other studies, we can use just a transgene probe and do serial section assays plus and minus RNA removal to show how much of the transgene probe signal corresponds to vector. Here's an example of RNAscope being used to verify and measure the tissue-specific expression of an AAV transgene. What you can see in the cardiac muscle and the skeletal muscle is signal that's distributed throughout the cytoplasm. Obviously, that's mRNA. In the middle panel, liver, we see only a few individual dots in nuclei corresponding to vector genome, indicating that the transcript, the promoter, is only active in muscle tissue, not active in liver. Therefore, we only see transcripts in muscle and not in liver. I showed you earlier in the presentation that our RNAscope and BaseScope assays are able to detect and visualize individual molecules of nucleic acid. In this case, detection of individual copies of vector genome, AAV genome, and transgene mRNA. With software such as the Halo software from Indica Labs, we can quantify those signals, numbers of vector genomes and transcription level on a per cell basis within an entire intact tissue. That's an application we apply quite frequently in these studies to understand the numbers of cells that have taken up virus and the expression distribution pattern within that intact tissue. Other softwares will do this as well, but our preference over time in the Pharma Assay Services Lab has been to use the Halo software. I mentioned very briefly earlier in the presentation our relatively new microRNAscope assay. This, as the name implies, was developed to enable detection of low copy endogenous microRNAs, but the majority of our work in the Pharma Assay Services group has been detection of exogenously delivered siRNAs and ASOs. Now, many of those studies have been looking at molecules delivered by vesicles such as lipid nanoparticles, but we've been starting to do more projects where these are being expressed off of an AAV vector. So just to show you a couple of illustrations of this type of assay, microRNAscope assays, these are assays detecting lipid nanoparticle delivered siRNAs, but they will be applicable to gene therapy studies just as well. So what I'm showing you here is simply a microRNAscope assay showing a very strong signal in treated mice and no signal whatsoever in vehicle-treated mice. So much like RNAscope assays, very clean assay, no background in untreated animals, very strong and specific signal in treated animals. Here's another example of a microRNAscope assay, in this case to detect an exogenously delivered ASO, also delivered in this case by lipid nanoparticles, but could just as well have been delivered by an AAV vector. So we're showing you here ASO signal in a treated animal, not showing you the negative result, which shows no signal whatsoever, like the siRNA assay shown previously. And then the consequence of ASO treatment is an exon skipping event. And what's shown in the middle panel and the right-hand panel are slides stained with a BaseScope duplex assay targeting an exon 1, exon 2 junction versus exon 1, exon 3 junction. In the untreated animal, there's only exon 1 visible as the sort of greenish blue stain. And in the ASO treated animal, there's now the appearance of the exon 1-3 junction. So again, these types of assays, in this case, were applied to a study of a lipid nanoparticle-delivered ASO, but could just as well be applied to gene therapy studies or AAV-based delivery of this type of molecule. What I've presented today are ACD's pharma assay services for gene therapy in situ analysis, with an emphasis on molecular quantification and morphology-based analysis. With RNA-scope, base-scope, and microRNA-scope assays, we can provide analysis services in which to analyze the biodistribution of any viral vectors, endogenous and or exogenous mRNAs, as well as RNAi molecules. An exceptional feature of our ISH technologies is the proven single molecule sensitivity that is greater than any other tissue-based analysis method. And in comparison to analysis methods like PCR of extracted RNA or DNA, the molecular analysis that we can provide with in situ hybridization is provided at a single cell level. Moreover, in multiplex analyses, we can provide further resolution of AAV biodistribution with cell type specific markers. And finally, I would like to emphasize that we can and do support gene therapy programs from very early stage to GLP tox studies and IND submissions. And even we have some projects where we're supporting clinical trials of gene therapies. We work with dozens of gene therapy companies in the US and in Europe, and I hope that what I presented today will give you reason to enable us to work with you as well. Thank you very much for your attention.
Thank you for that great presentation, Dr. Bunker. Now I would like to invite our second speaker, Dr. Chris Hager. Chris is the Senior Manager for the Application Science Group at ProteinSimple. Chris's group is focused on fostering both external and internal collaborations, create scientific collateral, and supporting various business units within ProteinSimple. Chris has a broad, in-depth knowledge of immunoassays of many flavors, capillary electrophoresis, and antibody design, production, and purification. Chris, the floor is yours.
Thank you, Miguel, for the introduction. My name is Chris Hager, Director of Application Science for the Analytical Solutions Division of Bio-Techne, here today to talk about the innovative capillary electrophoresis solutions for gene therapy research and development. Bio-Techne has a broad portfolio of manufacturing and analytical solutions for cell and gene therapy applications. These include a wide variety of GMP-grade reagents, including cytokines, growth factors, small molecules, and media, supplements and reagents for cell activation under GMP conditions, non-viral gene editing tools, antibodies and ISH technologies, immunoassays, and lastly, automated analytical instrumentation. Our innovative solutions all come with flexibility, customization, the consistency and reproducibility and scalability to support you from early stage through the clinic to patients. Today, I'll be focusing on our analytical solutions for viral vector characterization. Over the last several decades, there has been a tremendous evolution in therapeutics. There was a time where small molecules were the leading class, like Gleevec shown here. In more recent years, we have seen the increase in biotherapeutics, and this really started a few decades ago with humanized monoclonal antibodies. But today, while there are still a lot of new antibodies coming to market to treat disease, pharmaceutical companies have also realized and begun to leverage newer therapeutic modalities, including viruses, nanoparticles, and of course cells. The majority of current cell and gene therapies leverage two key viral vectors that can be classified by where they're used. For in vivo delivery, AAVs are leading the way. For ex vivo work, cells are typically extracted from the patient and modified with lentivirus to provide the cells an advantage in fighting disease in the body. There are certainly other viral vectors in development, including retrovirus, adenovirus, and HIV, but they're used currently to a lesser degree. AAVs are an attractive gene therapy delivery tool. However, they are also diverse and complex. There are 10 naturally occurring serotypes that infer different tissue tropisms or targeting capability of the AAV. A complete AAV particle is comprised of a 60-mer protein capsid made from 3 proteins, VP1, VP2, and VP3, that are present at approximately a 1:1:10 ratio. An additional layer of complexity to these proteins is the fact that there are over 50 post-translational modifications across these serotypes and capsid proteins. Lastly, AAV particles are approximately 4 megadaltons in size and can hold up to 5 kilobases of DNA. AAVs are also challenging to manufacture. Overall, the process is long and expensive. It's also hard to scale due to the complex nature of these biopharmaceuticals. As such, there is a lot of potential risk in the process. These challenges require manufacturers to carefully design and implement tests and control strategies. These tests are meant to address various attributes of the viral product. Let's take a closer look at viral vector quality attributes. For viral vectors, there are several key qualities and attributes that need to be addressed to understand and monitor. These qualities include identity, potency, purity, safety, and stability. The column on the right is a list of the techniques used to address these attributes. When I look at this list, I see a lot of tried and true but unevolved techniques. SDS Page has been around since the 1960s. Analytical ultracentrifugation since the 1920s, and even Western blotting at 1979 is a pretty old technique. Bio-Techne have built on some of these existing platforms and evolved them to a point to get faster insights and ultimately a better view of their product, summarized here. These platforms can be applied to help address these CQAs, including Simple Western, Simpleplex, and as I would like to discuss today as well, Maurice. Bio-Techne offers capillary electrophoresis instrumentation that can support the entire development path. If we think about the development process, we can start with cells and then go through a development process like this and end with pure AAVs. In early stage work, our Simple Western platform is ideal for not only discovery but also early key drug development work. As your product becomes more defined and pure, our Maurice platform can take over to support late-stage characterization, QC, and stability testing. The Simple Western platform is the new gold standard in Western blotting. It's our completely automated capillary-based Western blot platform that removes the laborious, notorious Western blot from your workflow, giving you back time and confidence in your data with high precision and reproducibility. Summarized on the right are the Simple Western advantages. These include fast time to results, with data being able to be ready in as little as 3 hours. The Simple Western platform offers both absolute and relative protein quantitation and features our built-in Compass analysis software. The Simple Western platform is quite flexible, offering various throughput options, able to run up to 96 samples in a run, and has multiplexing capabilities. The Simple Western platform is also quite reproducible with low inter and intra-assay CVs, and lastly is quite sample conservative, using very small sample volumes where you can start with as little as 0.6 microgram per mil or 3 microliters of sample per well to get picogram level sensitivity. Simple Western has multiple detection capabilities. The technology was founded on ultrasensitive chemiluminescence at its core. Here's a great example where we can detect as little as 0.4 nanograms per mil of CD19, the CAR T therapy target. But Simple Western has also evolved over the years and now includes two-color fluorescence as well. Here's a beautiful example looking at EGFR signaling with both total and phospho-EGFR antibodies in multiple channels on a JETS instrument. In A431 cells, without stimulation, the EGFR receptor is off and there's no phosphorylation. And this is seen in lane 1, where we have no signal with a phospho-EGFR antibody. However, in lane 2, we can see that there is substantial EGFR present in the A431 cells. Upon stimulation with EGF, The EGFR receptor is phosphorylated at multiple sites, and this is detectable as shown in lane 4, as well as in lane 6, where we multiplex the phospho and total EGFR antibodies together. In addition to chemiluminescence and the dual color fluorescence, we've introduced in-capillary protein normalization. This is a big one if you want to publish a paper. Loading controls just don't cut it anymore. So assessing the total protein is a must, and we figured out a great way to give you that in the exact same capillary as your immunoassay data. Since Simple Western is an automated platform, the reproducibility is outstanding. Whether you're looking within a run, like this PI3 kinase CDK6 multiplex, where we have CVs for 24 replicates under 10% for each target, or day-to-day and user-to-user, like here on the right. 4 different users, 4 different days for one great week of data with CVs under 12% per day, and overall under 10% for 4 days and 44 replicates. To show how the Simple Western platform is being applied to viral vector work, I've summarized a collaboration between ourselves, the Cell and Gene Therapy Catapult, and ProGen. In their work to further improve the development and manufacturing of ATMPs, the Cell and Gene Therapy Catapult used Simple Western to monitor AAV2 purification. That work was summarized in an application note shown here. Since WES only takes 3 hours from start to finish, the Cell and Gene Therapy Catapult can quickly get their data and make better, more accurate, and faster decisions on process improvements. And AAV2 purification was first looked at using our total protein assay, which you can think of like your Kumasi stain. With this assay, all of the proteins in the sample are visualized, and over the course of the purification, we see fewer and fewer bands until finally the 3 main viral proteins are shown. With this assay, since you're looking at the total protein, it's not always easy to follow the viral proteins in the dirtier, earlier stage samples. However, the Cell Gene Therapy Catapult also ran the immunoassay on WES using an anti-VP specific antibody from ProGen. In this run, essentially only the VP proteins showed up. So it's a lot of data generated in a really short amount of time, providing deep insights into the purity and identity of your samples. Those data come with a lot more detail as well. Shown here is the same elution fraction detected with the total protein assay in the top panel or immunoassay in the bottom panel. The immunoassay misses a key impurity in this fraction since it isn't VP related. Lastly, in the application note, we went back to a purified AAV2 sample and interrogated it with several VP-specific antibodies from ProGen to further demonstrate the utility of the assay to identify and track individual capsid proteins. This past month, we released a new application note taking the analysis of AAVs on Simple Western further on the JESS platform. The application note, entitled Concentrating on AAV Impurities with Ultrasensitive Total Protein Detection on Simple Western, features the JESS platform as well as our replex. For this application note, we're again leveraging our total protein assay, but we've turned up the sensitivity by increasing the concentration of our labeling reagent. The application note also uses our replex feature available on JESS as well as the AAV instrument. Replex you can think of like strip and reprobe, but much better, as well as completely automated. In this application note, we use the replex feature to look at impurities in AAV process samples. We use the same AAV antibody to look at VP1, 2, and 3. And we follow this up by replex and total protein detection. We looked at AAV1, AAV4, AAV7, and AAV9 with this approach. The assay only required 50,000 copies of AAV per sample well, which is great considering how challenging it can be to get enough AAV to work with. And as expected, the antibody reacted quite well with AAV7 and AAV9, but not AAV1 and 4 serotypes. The data can easily be quantified for both VPs, shown on the top, or for the total protein, shown on the bottom. And certainly, the total protein data can be used for any normalization you're interested in between the samples. We next sought to compare the 5X approach with CyProRuby, which is used frequently with SDS-Page for detection of impurities and other low concentration materials. CyProRuby can detect typically 1 nanogram of protein at the lower levels of detection. For this experiment, we use a commercially available AAV2 sample of 1 × 10⁻³ GC per mil and serially diluted it from 1:5 to 1:40 before analysis on SDS-Page, followed by CyProRuby staining and imaging on our fluorochem imager, shown here, or analysis on Simple Western. Comparing the results, Simple Western can easily detect the same amount of protein as CyProRuby and is actually more sensitive. Shown in this middle panel is the same 1:40 dilution for both the CyProRuby gel image shown on the top and the Simple Western data shown on the bottom, showing better detection at the same dilution. And going further, we were able to obtain quantitative data using as little as 160-fold diluted AAV2 on the Simple Western assay. Our Maurice platform is a dual-mode instrument providing the gold standard image CIEF technology and CESDS. These two techniques are widely used by biopharmaceutical industry to characterize therapeutic proteins, and we see a lot of advantage in the gene therapy space as well. The platform is easy to use, comes with CFR Part 11 compliant software, and has Empower Control access as well. Maurice SDS provides a widely accepted platform for biopharmaceutical analysis. At only 35 minutes per sample and up to 48 samples per batch, CESDS on Maurice is a fast way to get the purity data for your AAV samples. As I'll show you, the data is reproducible as well with RSDs typically under 10%. Maurice is also really easy to use, whether you're a new user or an experienced bench scientist, making it a great choice for any user from R&D to QC. We've generated a nice application note where we've used Maurice CESDS to look at AAV sample purity. Let's take a closer look. As I showed earlier, SDS-Page is traditionally used for viral vector identity and purity, but is a really old technique. Anyone that has run a gel can attest to some of the challenges of the method. When we first purchased a commercial AAV2 sample, this is the SDS-Page that came with it. The 3 viral proteins can be seen, although somewhat weakly, and there are also some other bands present in the sample. With Maurice SDS, the results are much clearer. Using only a few microliters of sample for analysis on Maurice, here's our new lane view of the same sample, much nicer looking. Behind that is a data-rich electropherogram, which we can use to accurately measure the purity of the sample, which in this case was only approximately 70%. We use the same AAV2 sample for further characterization of our method. We ran 5 replicate injections of the AAV2 sample and measured the % peak area and capsid ratio for each. Overall, the corrected % peak area measurement was very precise, with all RSDs under 4.3%. The observed capsid ratio for the sample was approximately 8:1:1, again with really low % RSDs, 6.9% across the replicates. We also have examined preclinical AAV protein purity with Maurice CESDS. The method was able to identify a total of 11 peaks in 3 of the 4 AAV samples we looked at. These include 2 low molecular weight species, the 4 proteins we're normally expecting to see, and then 4 additional high molecular weight species. The method can quantify a 0.1% impurity, and overall purity values for each sample were assessed. In addition, the method was able to detect an additional peak in sample 2, highlighted here between peaks VP2 and VP3. I'm now going to move on and spend the remainder of my time talking about our IEF methods on Maurice. Maurice is also a great platform for characterizing your AAVs by charge. Maurice provides the gold standard for charge heterogeneity analysis of biopharmaceuticals and is widely accepted and used. Unlike ion exchange chromatography, IEF is fast at under 15 minutes per sample and has high throughput capabilities, able to run up to 96 samples in a batch. With Maurice, you also get detection flexibility. Maurice ICIF uses 280 nanometer light to provide absorbance data, but is also equipped with both native fluorescence and an optional blue fluorescence detection capability. We have leveraged all three detection capabilities for AAV analysis. The ICIF methods are also reproducible with RSDs under 10%. We've published an application note, shown here as a snapshot, where we describe some of this work. You might ask why one would use ICIF to analyze AAVs. It has previously been reported that not only do the individual capsid proteins have their own isoelectric points, but the assembled capsids do as well. This was nicely reported in the Journal of Virology as shown here. In the graph on the right, notably most of the individual proteins as well as the capsids have PIs around 6. Our imaged IEF technology is the industry standard for examining charge heterogeneity. This can be done for therapeutic proteins, large, complicated fusion proteins, or monoclonal antibodies. And so we've applied that technology to look at AAVs and leverage the differences in isoelectric point that I was showing you earlier. For AAV analysis using ICIF mode of Maurice, we have developed two different methods that can be used to characterize AAV capsid proteins or intact AAV particles. These methods have different approaches and goals. The capsid protein method breaks the AAV capsid into individual proteins using a denaturation approach, while the particle characterization method is much more mild and maintains the AAV particle while providing sufficient solubility for the analysis. Both methods can be run in under 12 minutes. Both only use a few microliters of AAV sample at 1 × 10⁻³ GC per mil. Both methods leverage the absorbance and native fluorescence of Maurice. Shown here are examples of native fluorescence images from Maurice, looking at an AAV6 sample in triplicate for both methods. For the capsid protein method, we clearly see 3 peaks, one for each of the capsid proteins, close to the expected PI of the proteins. For the particle method, we see the same sample with a significantly higher basal PI, a reduction to essentially one main peak with a small shoulder. Let's see how these methods can be used. We first looked at a series of preclinical AAV samples with the denatured method. These samples were normalized by VG or protein content before analysis. We found 15 unique peaks in these samples in this beautiful pattern. These peaks were quantified, shown on the right. This is a lot of data, so I boiled it down to the acidics, mains, and basics like we use for biotherapeutic analysis. There are subtle differences between these 5 samples, so it is interesting to start looking at trends using the denatured method. We've examined the use of the ICIF methods for stability testing by taking the same AAV6 sample and incubating it at various temperature conditions. To induce a rapid, noticeable difference with the capsid protein method, the AAV6 sample was heated to 95 degrees C for 5 minutes prior to denaturation and analysis on Maurice. The differences between the reference sample, shown in blue, and the stress sample, shown in orange, can easily be quantified using our COMPASS software. We see increases in several acidic species and decreases in the main and basic species for this AAV6 sample. which indicates that this method is suitable to examine small changes from heat stress. AAV particles have a melting temperature at which they naturally fall apart. This is typically somewhere between 60 and 80 degrees C, depending on the serotype and the duration of the heat exposure. We incubated the AAV6 sample at either 37 degrees C over the weekend, 50 C or 60 C for 10 minutes, and compared them to a reference sample using the particle method. We saw that the AAV6 was quite stable, remaining unchanged over the weekend and remaining intact at 50 C for 10 minutes, but disappearing after incubation for 10 minutes at 60 degrees C. These data together show that the particle characterization method and the denatured capsid protein method are useful for assessing AAV stability. To establish Maurice for empty-full AAV characterization, we again collaborated with an industry leader, this time Ultragenyx. This collaboration had its first public data presentation in December 2020 in a webinar hosted by the Cell and Gene Therapy Insights. Here's a QR code that links directly to the webinar if you want to get it now. Take a picture. I'm going to highlight the key findings from this collaboration thus far. The theory for using an intact method to distinguish empty and full AAVs is that an intact AAV containing DNA has a different overall charge or shape that's different from an empty AAV. We started by looking with Ultragenyx at intact AAVs with limited additives and avoided denaturants. The first thing shown in the Ultragenyx webinar was that additive screening was employed to find the optimal separation conditions. In developing this intact method, several additives were screened for optimal reproducibility, resolution, and separation potential for empty and full. Shown here is a snapshot from the development where additives 1 and 2 were screened against a panel of AAV samples and compared to the combination of both additives together. In the end, additive 2 produced the most desired result from the criteria defined by Ultragenyx. Similar to what we had shown previously in the application note, stability testing was carried out using this new method to assess its capability to identify stress peaks. In this particular case, an AAV sample was incubated at either 40 degrees C, 50 degrees C, or 65 degrees C and compared to a reference sample. An increase in acidic species was seen in the higher temperature samples, which can be quantified as shown below. The next point from the Ultragenyx webinar was that leveraging UV and native fluorescence on Maurice provided powerful insights into the empty and full status of AAVs. When the absorbance, which is a combination of signal from DNA and protein, and the native fluorescence, which is protein only, were compared for full, intermediate, and empty samples, we observed some very interesting differences. The full and intermediate samples have substantial UV and native fluorescence signals, while the empty sample has very little UV signal. However, when we look at the native fluorescence, we see a strong signal for the empty. Comparing UV and native fluorescence therefore appears to be quite important for distinguishing empty and full AAVs on Maurice. To further show that using Maurice's dual wavelength detection helps elucidate empty-full status of AAVs, we analyzed commercially available empty-full samples with an in-house method. Shown in blue is the absorbance of the AAV9 full sample. In black is the native fluorescence, which again is looking at the protein signature only. There's significant overlap between the absorbance and native fluorescence for this sample. However, when we look at a nearly empty AAV9 sample, we observe significantly less absorbance, shaded in orange, and in this case, a substantial native fluorescence signal again. These native fluorescence signals from the two samples can be quantified, shown here on a per peak basis, and then in the last column as a sum of the two native fluorescences. Overall, this shows that the native fluorescence between the two samples is essentially the same. However, if we quantify the absorbance between the samples, there's approximately an 8-fold difference, which is fairly close to what was reported by the manufacturer with a different method. The last key point from the collaboration with Ultragenics was that using Maurice's 458 nanometer filter and a DNA dye could provide another path to characterization of AAV empty and full samples. Maurice has several filters that can be leveraged for AAVs, as I've already started to describe. These include the absorbance and native fluorescence channels, but as I'd like to now focus on the optional 458 nanometer filter. In this work, we're using the native fluorescence channel and our 458 nanometer filter and a DNA binding dye. This DNA dye binds to the DNA inside the AAV and results in what we've called the extrinsic fluorescence. Both filters and the UV absorbance can be collected for each injection, and there's minimal crosstalk between the 458 and native fluorescence channels. The application of a DNA dye that fluoresces at 458 nanometers to a full, intermediate, and empty AAV8 sample are shown here. The protein signatures from the AAV are shown in black from the native fluorescence. In blue is the extrinsic fluorescence from the DNA dye complex and the 458 nanometer filter. The empty sample has nearly the same native fluorescence as the other samples, but no 458 signature, suggesting that it has, again, no DNA inside of it. I would like to summarize today's technical seminar by bringing back this slide and populating it with data from today. We started upstream in the process with Simple Western to characterize AAV purification fractions, and then moved downstream and looked at pure AAVs by CESDS and denatured AAV capsid proteins by charge heterogeneity. We also looked at an intact ICIF method to characterize empty and full AAV particles. We are excited to be able to help support the entire R&D pipeline and look forward to our continued collaborations and business with all of you. And with that, I will be happy to take a few questions. Thanks.
Thank you, Dr. Hager, for the presentation. Before we begin the panel discussion, I'd like to remind attendees that they can submit questions to the panelists using the Ask a Question box in the GoToWebinar control panel on the right side of your screen. All right, so now it's time for the first question. We have one question for Dr. Hager. I wonder, what are the anti-VP1, 2, 3, and different serotypes of AAV? Or are they different antibodies developed specifically for capsid proteins in each serotype?
Sure. So the VP proteins for each serotype are very similar but slightly different. So the VP123 antibody that we used, that I showed today, is actually made from immunogens from 3 different serotypes, I believe. So it does recognize multiple serotypes. It doesn't recognize all of them, as I showed today. So in certain cases, you might need a different, more specific antibody for your serotype, and there are other ones available.
Got it. Great. Thank you.
Sure.
Next question is for Dr. Bunker. What are the catalog probes, or what catalog probes do you have available for AAV, and how would it get a custom probe made?
So we have quite a few probes for AAV. Obviously, the vectors vary a lot from one customer to the next. We have probes to many of the common AAV promoter and enhancer regions, also to essentially all of the standard reporter genes. We have probes to both the coding region, positive strand, as well as negative strand for DNA detection. And for custom design probes, it's typically quite straightforward, depending on the species being where the probes are being used and whether or not the transgene is normal human or codon optimized. That will determine whether or not we use RNA scope or base scope. But from the point of being provided with a sequence for either the vector or transgene to having a probe design, it's typically a couple of days, and then it's maybe about 2 weeks from that point when you have a probe that's ready for use.
Got it. Perfect. Thanks for that answer. And this next question, I believe Emily answered it in the chat, but I thought I'd bring it up to everybody. Can RNAscope be used with cell suspensions?
So it can be used. Oh, go ahead, Emily.
I can also reiterate the answer I gave in the chat. This assay really is designed as a slide-based approach for analyzing RNA, whether it's in tissue sections, or we do have tech notes that are validated by ACD for use in cultured cells. Oftentimes people are using chambered slides or growing cells on cover slips. And then we also have tech notes for using cytospin if you have a non-adherent cell population. This works in a 96 well plate, but you know, it really is designed as an approach for tissue or cells that are anchored onto something, if that answers that question.
Yeah, so in the context of, there are quite a few studies where we're actually, more oftentimes we're looking at lentiviral vectors or transgenes in bone marrow biopsies or in PBMCs, and in both those cases we will prepare cytospins for analysis.
Perfect. Thank you for that answer. This next question is for Dr. Chris Hager. How would you compare Maurice CE-SDS versus PA800 plus or AAV?
Yeah, sure, that's a good question. They're actually quite comparable in data I've seen from the P800 plus. It's pretty close to what we get. I think the benefits we have is a much quieter baseline. The system's way easier to use as well. So comparable data, slightly better resolution, I think, as well, but faster data and easier to handle. So some good benefits there.
Got it. And while I have you on, there's another question. Could you comment on the sample prep procedure for the denaturation of capsid proteins?
Sure. So the denatured ICIF method, we mildly heat the AAV in the presence of organic solvent, DMSO, for a few minutes, and that seems to well disassociate the viral protein. You can find more details about that in the application note on our website.
Perfect. And can you also determine the EF ratio of an AAV sample at intact capsid level with Maurice?
So EF, the empty full. Yeah, I think we're at a point where we're seeing really interesting differences, especially when you leverage the absorbance and the native fluorescence. And in the example that I showed, compared to what was provided by the manufacturer, we were actually pretty close to the full difference in the DNA that we calculated with our technique. So I think we're on that path. Certainly more work needs to be done, but really encouraged by what we've shown so far.
Got it. And we have one more question for you, Dr. Hager.
Sure.
Can the 458 nanometer filter be installed on an existing Maurice instrument?
Yeah, absolutely. So that's available on brand new instruments if you want to order it with one, and also available on existing instruments. Just reach out to your local sales rep and they can help you get started on that.
All right, perfect. This next question is for Dr. Bunker. How can I differentiate AAV vector DNA from mRNA?
So we take a few different approaches. Again, I emphasize the fact that a probe to, say, a transcriber region will detect both the vector, the transgene itself, DNA, as well as the transcript. There's no way to make that probe recognize one but not the other. So the various approaches we take, on the one hand, as I showed a couple of examples, we can simply look at the amount of signal that comes from a probe that detects only the vector and compare that to the amount of signal coming from a probe that is targeting both vector and the transcript. And typically we'll see a pretty significant difference, the vector probe generating just a few dots. the other approaches we can take is one is an RNA removal approach or a DNase approach to remove one type of nucleic acid to the other. And the third approach is one we do occasionally, which is if your construct has a synthetic intron in it, we can use the base scope probe and position oligonucleotides on either side of that intron, such that signal will only be generated when the intron is spliced out, and that will give an RNA-only signal. So those are kind of the three approaches we might take.
Perfect. Thanks, Chris. And we have one more question for you, Dr. Bunker. Can you combine an AAV probe with a cell marker, either with using RNAscope or even looking at the protein with an antibody on the same tissue section?
Yes, so combinations of probes to AAV, vector, transgene, and anything else for that matter is very straightforward. All of our probes can be combined without any sort of assay development, and you can detect essentially any transcript encoding a cell marker with RNAscope or base scope. So combining an AAV probe with cell markers, whether it's from neuronal tissues or liver or kidney or any other for that matter, that's very straightforward. We've also fairly recently developed methods that make it more straightforward to combine IHC or immunofluorescence with RNAscope, so that using well-established cell marker IHC or immunofluorescent assays with RNAscope for AAV detection, that can also be done with a little bit of development work.
Got it. And while I have you on, there's one last question. Is there any quantitative measurements for RNAscope technologies, or are they strictly visual assays?
So I didn't spend too much time on it, but we have quite a bit of information on our website, including some nice webinars that illustrate the use of image analysis software, which will effectively quantify signals on a per cell basis. So we like to emphasize that in comparison to, say, a qPCR assay, where you take a tissue and extract the RNA and turn the entire tissue into n=1 data point, with in situ hybridization, you're getting data on literally tens of thousands, if not hundreds of thousands of cells in an intact tissue, and quantifying the abundance of a message or copy number of vector in each individual cell. And that's, again, using image analysis software, which will count dots or measure total signal on a per cell basis.
Got it. Thank you, Dr. Bunker. This next question is for Dr. Hager. Could you comment on the intact analysis method in terms of extrinsic fluorescence for empty versus full analysis? Do you add the dye to the sample directly, or are there other steps involved?
Yeah, so unfortunately, I can't really discuss at this time anything further about how that technique is achieved.
Got it. And we have one more question. How many viral copies are needed per well for detection?
Is that question for me also?
Yes, that's for you, Dr. Hager.
Gotcha. I guess it really depends on which technique we're talking about. With our enhanced total protein on Simple Western, we were down to, in a 3 microliter sample, we're talking 50,000 copies at the lower limit of detection for that assay. Because Maurice and CES, and ICIF are both direct detection methods, the sensitivity is a little bit more dependent on the absorbance direct measurement. So I think typically with both CES, and ICIF, 10 to the 11th, 10 to the 10th is kind of the lower limit of detection currently.
Got it. Thank you. And there's one question on drug development. Which instrument could they use for drug development purposes?
So as I showed that pathway, I actually envision and wholeheartedly believe that Simple Western and Maurice together support the entire development path when you start out early in R&D, and then when you have a final purified AAV product that you're trying to release lots of. So I actually think they're the same techniques at their core, size and charge separation, just one's coupled to immunodetection, one's not. So they're very similar and they work very well together. And a lot of our biopharmaceutical customers working on monoclonal antibodies, for example, have already realized the benefit of having both.
Got it, perfect. And there is another question for you. I am researching the identification of phosphoisoforms with the same antibody that detects the total species. Which instrument would you recommend?
Sure. So if you're looking for phospho-specific detection of proteins, I'd probably, well, I mean, you can go a couple ways. Certainly our charge separation on Simple Western with a specific total antibody will show you those phospho peaks because phosphorylation induces a charge change. You could do that on a size separation too, and something like our JES instrument, you could do 2-color fluorescence, like a total and a phospho together on the same target, like I showed for EGFR today. There's a lot of different ways you could cut that and achieve that goal.
Got it. This next question is for Dr. Bunker. Can I use the same probe for chromogenic and the fluorescence assay, or are probes used for one or the other assay?
Yeah, so it's not entirely clear from the presentation sometimes, but the probes themselves are not labeled. It's the detection reagents that determine what your method of detection is. So you can either use either HRP or alkaline phosphatase or various fluorophores with the same probe.
Got it. One more question for you, Dr. Bunker. Can these assays be run in the lab as well?
Oh, certainly, yeah. So most of our customers use RNAscope or BaseScope or microRNAscope in their own labs. We have assays which run with a benchtop hybridization of it as a manual assay, and we also have the same assays can be run either on Leica Bond RX or Ventana Discovery Ultra instrumentation. So by all means, we can support and enable you using these assays in your own lab, or you can outsource to us.
Perfect. Thank you, Dr. Bunker. This next question is for Dr. Hagar. It's an excellent presentation, Chris. Do you offer services for phosphoprotein quantification?
So services makes me think you want us to do it for you, which we absolutely can do. So we do offer custom Simple Western and Maurice, while I'm saying it, assay development and method development. Perfect. So feel free to reach out, and we can definitely talk about what that project might look like.
Got it. And this next question is on the Maurice. Do I have to specify which mode of detection I want before a run?
So in between CESDS and ICIF, 2 things dictated. Actually, the cartridge itself is most of it, of course your reagents as well, but the software knows which cartridge you're using and won't let you run the wrong type of assay. And then the software, of course, has the appropriate batches for execution of the run. When it comes to detection of absorbance versus native fluorescence, that's automatic in terms of ICIF. You get both at the same time, although you could change the native fluorescence exposures if you like. And similarly, the same thing would go for the 458 if you have that installed.
Perfect. And while I have you on,
sure.
There's one question for you. It says, my AAVs like to aggregate. How can I keep this to a minimum?
I don't think your AAVs are unique in that phenomena. Yeah, so it's something that we definitely had to work on in our intact method. So I think the key, and I highlighted on that slide that Ultragenics showed as well, mild solubilizers to keep the AAV happy and from sticking and aggregating, but not so much that you make it fall apart. So mild solubilization, typical solubilizers for ICIF would apply there. So.
Perfect. And we have one question for Dr. Bunker. Can the RNAscope assay be used to multiplex beyond the 2, 3, or 4 plexing capabilities that you showed in the presentation?
It can. We have actually it can be multiplexed to a quite high level. We have an assay called HighPlex, and we have HighPlex Up, which is both of those enable a multiplexing to either 12-plex with HighPlex, and with the HighPlex Up, I think we can maybe go to 48-plex, something kind of extreme. And that involves doing iterations of either 3 or 4-plex with fluorescence scanning in between. We develop methods for decoupling the fluorophores and then coming in with another set of fluorophores for the next set of probes. But so yeah, up to 12-plex is quite straightforward with the HighPlex assay.
Perfect. And we got one question for Dr. Hager. You showed nice AAV data on the Maurice, especially with intact AAVs. Is this method applicable to other serotypes?
It's a good question. So yeah, the work that I summarized from what Ultragenics showed was with AAV 8 and 9, and then what I showed also was AAV 9. Yeah, certainly I think the general principle of, like I mentioned, solubilizing, not denaturing, that's something to keep common. As I showed from that summary of the virology paper, most of the serotypes should have around the same isoelectric point. So that would mean the same or similar pharmalytes or separation amphilytes could be used. I don't think that every single method will work. It's not fit for purpose for every single serotype. It's probably needs a little bit of personal method development, but I think we've got some great starting points for you.
Got it. Okay. And then this question's on the Maurice. How many detection channels does the Maurice IEF have?
Yeah, sure. So the default, again, is the absorbance and the native fluorescence, which looks at the tryptophan native fluorescence, and then the optional blue or 458 nanometer fluorescence, so 3 overall.
Perfect. This next question is for Dr. Bunker. Aside from the RNA scope assays, do you have any to detect RNA? Do you have any assays to detect also DNA, either viral or non-viral DNA?
So we, as it turns out, the RNA scope assay is very effective at detecting most any extra chromosomal DNA, whether it's a plasmid or any sort of virus or vector. That type of nucleic acid is generally quite accessible to the oligos under the assay conditions for RNA scope. We recently developed and launched a formal DNA scope assay, which uses different pretreatment conditions which expose and make accessible genomic chromatin. And so that was developed with the intention of enabling detection of genomic alterations in the context of oncology, but it would also be very useful for looking at integration of gene therapies that are introduced by homologous recombination or using other vectors like lentivirus.
Perfect. Thank you, Dr. Bunker. And so we've actually running out of time now, so we'll end with that answer from Dr. Bunker. If we didn't get to your question, we'll reach out to you after the webinar and answer it directly. I'd like to thank our speakers today. There was 2 great presentations by Dr. Chris Bunker and Dr. Chris Hager. And I'd also like to thank everybody who attended this webinar today. We really appreciate you taking the time out of your busy schedule and hearing our presentations.
Yes, thank you all for attending.
Thanks so much.